Hello dear students, how are you doing? I hope you are doing well. So for today's lesson we are going to cover two main topics. First topic we will study about DNA replication. Second we will study about gene expression, which has two steps, transcription and translation. So let's get started with the DNA replication, or we can say DNA replication is the beginning of cell division. Do you remember we studied cell division in chapter 1? Last part of chapter 1 we have cell division, and we have two types of cell divisions. We have two types of cell division, mitosis and meiosis. With the beginning of cell division we need DNA replication. Forward, DNA replication is important for duplicating itself and having two different cells. Okay, and we can summarize DNA replication steps to the three main steps. So the replication of DNA has three main steps. Now I'm going to summarize it for you and show you on the figure also. First, it says helicase enzyme separates DNA chains. So just imagine a helicase enzyme works like a scissor. How you are going to cut and separate a stuff, okay, an object. Also helicase enzyme starts with breaking, okay, breaking and cutting the bonds, which is they are cutting the hydrogen bond between nucleotides and separate two strands of DNA. Just imagine like this. If you have DNA like this, okay, like so, there is a hydrogen bond between nucleotides. So when we have DNA like this, it starts from here, helicase enzyme, and starts to cut the bonds. Okay, and after this we have the second step says, or before that one, it says the place which bonds and nucleotides has been separated, it looks like Y shape. Later I'm going to show you. That's why we call this place replication fork. It looks like a fork. Step 2 says enzyme of DNA polymerase add complementary nucleotides. Step 2, DNA polymerase enzyme collects the nucleotides and make the complementary strand. Okay. Because we have three nucleotides inside cell, of course, and where there is a base strand base, a base chain, so the complementary one will be stand on it. Last one says DNA polymerase enzyme ends the replication. So as a result, we will be having two new strands of DNA. They are identical, and we call them semi-conservative replication. So if I show you the picture and the steps on this figure, what do we have? First of all, we have helicase enzyme starts to cut the bonds, breaking the bonds of nucleotides. We call them hydrogen bond, and the strands of DNA will be separated as we see. And after this, by the help of DNA polymerase enzyme, collects free nucleotides. You see? They are free nucleotides inside the cell and standing and, let's say, locating on the, let's say, nucleotides. So each nucleotide strands on it let's say, complementary base pairs, and we will be having a new DNA strand. So as a result, we will be having two new identical strands, but you should know that one strand is the old one and the second complementary strand will be the new one, but they will be almost the same. And there is something else, please pay attention. Always the direction of initiation, the direction of DNA polymerase enzyme will be opposite, in opposite directions. So any remaining parts which, let's say, direction of initiation and adding nucleotides will be opposite, and there will be some parts remains, some holes. Of course, it will be filled by DNA ligase enzyme, So it will fill the gaps and the complete strand will be done. So the second topic we will study about transcription of RNA. It means making RNA by using a single strand of DNA. Just imagine that. In DNA replication, it was very simple. We said DNA will be replicated and double itself. But there is another step that is a part of gene expression. A beginning of gene expression is transcription. So transcription is building operation of RNA by using DNA chain, a single strand of DNA. First step says RNA polymerase enzyme will attach to a part of DNA, and the second step says RNA polymerase enzyme adds free nucleotides and collect them and attached with the strand of DNA, and a part of RNA will be made. Okay? We have this example, even in Wuzari questions, in ministry questions can be asked. The question says, if the succession of the base of DNA chain is CAG and CTA, which is the, let's say, strand of DNA like this. The complementary base pair, of course, it will be RNA in transcription like this. So, can you tell us, are the complementary nucleotides of RNA? Of course, we should know that the succession of base pair of RNA will be this chain. For C, we'll be having G, cytosine guanine, adenine will be uracil instead of thymine. We said thymine can be found only inside DNA, but uracil can be found only inside RNA. The third one is G, will be cytosine. Cytosine, let's say guanine, thymine. Why do we say thymine here? Because it is DNA. Please pay attention. And this is RNA. So that when RNA will be made, it means it's ready and it will be released by RNA polymerase enzyme. We have this figure to show you how the base pairs are attaching each other and a strand of mRNA will be produced here. So by using a single strand of DNA, one RNA will be produced. There is a question. You are going to think, so when a part of DNA is separated, just like this, imagine. Let me show you. Here is a DNA. Okay, in this part will be separated DNA and RNA will be produced here. Maybe you are going to say, what will happen when RNA will be free? Of course, the two strands of DNA again, they will recombine and they will be doubled again. They will be. The third topic we are going to cover is translation. The aim of translation is making proteins. So we should know that after mRNA has been made by the help of transcription process and by the help of using of one strand of DNA, now we will be having protein synthesis, which is translation. So translation has three main steps. Step one is initiation step or stage. In here, tRNA connects with mRNA, which also there's an enzyme which connects by amino acid, which is methionine. This methionine is this amino acid. So this methionine regarded as the first nearly to all polypeptides, but it may disappear later. It is the beginning of collecting amino acids. So we said that amino acid is the building blocks and the basic unit of proteins. The stage 2 we called elongation stage. What does that mean? In this stage, polypeptide form anticodons. What does that mean, anticodon? Let me show you. For example, in anticodons, in here, we have three amines. Okay, in the third stage, which we call termination stage, when the ribosome arrives to the stopping codon like UGA or UAG or UAA, so the mRNA, this leads to separation of polypeptides chain, which was formed from the last tRNA, and release happens in the cytoplasm, which is leaving tRNA. Okay, so ribosome separates from each other and ribosome moves away from mRNA. So we have transcription and translation. So we already mentioned about transcription. We said by using a strand of DNA and by adding free nucleotides, so pre-mRNA will be produced inside nucleus. So the mature mRNA will be free and released to the cell, to the RNA. With the help of mRNA, they will attach with the ribosome. For what? For collecting amino acids. You see, these small circles are amino acids. If a growing amino acid chain by 3 amino acids, they will make 1 protein, as we have in this example. So after this, what will happen? By collecting a number of amino acids that we have in this picture, translation process happens. The aim of translation process, we will be having a newly formed proteins, which we call a mature protein. Thank you so much for watching this lesson. I hope it was helpful and you are going to get some benefits, and see you in the next videos.